we will go. Done. Okay, now Jasper's already won the Freddo Frog for the day. He's told me the significance of the staircase in Gattaca is, of course, but he hasn't. Jasper's at level 1 in terms of the significance of the staircase. Sure, it represents a DNA helix. Yeah? You've got this double strandedness. Now, and you've got these steps that go along. And DNA looks remarkably like this, because you've got this pairing across these stairs. But the real significance in Gattaca in terms of the staircase is who lives on the up top.
Jesus.
Wasting time. That was actually in the video that you watched. Who lives up the top? Vincent, the main guy. Yes. Now Vincent, his name means warrior or braveheart, and he wants to be an astronaut, a navigator, lead the way for humanity, if you like. And one of the things you hear is reach for the stars, you know, in terms of reaching for your goals. So down here is his friend in the wheelchair who's crippled in body but crippled in mind because he only thinks in terms of genetics and assigns his worth in terms of his DNA structure. And then he is trapped down here. So the Earth, if you like, is, you know, very pedestrian, and up here, you know, this aspirational level is where Vincent lives. And he is above DNA. See, all of the people running around in the movie focus solely upon DNA as a means of judging if someone is a worthwhile human being. And Vincent himself is above that idea. Okay? So the movie itself is fantastic for things like that. Now, as far as key learning intentions go, Watson, Crick and Franklin are the co-discoverers of DNA as the molecule of inheritance, but Watson and Crick got the Nobel Prize and Franklin did not, which is kind of sad, and we'll talk about that. To describe the structure of DNA and how it replicates. How does it do its job? To be able to outline the factors that contribute to causing mutations. If you've done your homework, which you should have done, there was that question 3AI, I think it was, where you wouldn't have been exposed to changes in DNA being called a mutation. And what causes mutations? We need to cover that key point as well. Now, there's a lot. These are your key points. Fine. Watson, Franklin, Crick, monomer, polymer, central dogma, biology, DNA, nucleotide, phosphate, sugar, nitrogen base. Hey, actually, put 5C sugar and highlight that, because you need to know that. Chapter and verse. You just need it. Okay? 4 nucleotide monomers. Oh, fine. Look, I can hear you quietly judging me back there. Fine. 5 carbon sugar. That's gone out there.
I was judging you.
Side, we'll move it up. It's not like that.
Make it look small. Oh, Jesus close.
Done. Okay, that is better. 5 carbon sugar, because if you write sugar, you'll be graded as incorrect. Okay, so a 5 carbon sugar, a nitrogen base, G, A, T or C, and phosphate. That is the atomic structure, if you like, of DNA. And not quite at the level of atoms. It's a phosphate group, it's a 5 carbon sugar, and your nitrogen bases. And here's like the staircase and your G's, A's, T's and C's. And those G's, A's, T's and C's, those nitrogen bases are the information that's being passed to the next generation. And it's the information that codes for proteins. Remember, DNA does 2 really important things. Codes for proteins, passes that information to the next generation. And I've mentioned before, you're just a big bag of proteins. Everything you are and everything you're doing, and the tools you're using for thinking and what not are just proteins. A lot of different proteins. 4 nucleotide monomers. Sugar, 5 carbon sugar phosphate spine. I'm going to change that as well. The DNA helix, complementary base pairing.
I think I need a diagram.
Oh, you do too. Just come here. Just here.
Hey, you're on.
Done. I've got that one. There you go. Complementary base pairing, DNA replication. What's going on there? Now, we're not going to... And, of course, all of this as well, which is on mutations, although you'll never be tested on these sorts of things. I'll change those to blue at some stage. Now, okay, you don't need to know about Chagaf, but he sort of set the stage. In the 50s, there was this big competition, this big drive to try and work out what was going on with DNA. Sorry, not what was going on with DNA, what was going on with inheritance. How was it that information is passed to the next generation? And some people thought it was proteins, and some people thought it was this or that. Something in the cell is doing that job. But what is it and how is it doing it was open for debate at that stage. Now, Chagaf showed the DNA, there are equal amounts of the nucleotides, adenine, thymine, as well as equal amounts of guanine and cytosine. Okay? So he said, look, the T's and the A's, always, it doesn't matter what species of plant or animal you look at, they're always the same amounts, and C and G are always the same amount. And so he knew something was going on, but that's as far as he got. Now, Rosalind Franklin took this particular, using X-ray, so it's an X-ray crystallography, so you've got to crystallise whatever it is you're trying to take an X-ray of, because X-rays go through soft material, and she managed to take this particular image, which to you and me doesn't look that significant, okay? But she successfully used X-ray crystallography to photograph DNA, okay? Franklin also determined the nucleotide bases were inside the helical structure, not outside, as Watson, Crick and Linus Pauling believed. Linus Pauling is this guy you've never heard of, who's won 2 Nobel Prizes in 2 different branches of science, which is extraordinary, and was looking at his 3rd.
What's mine?
Okay. And he needed to get to this particular conference where he was American, where he would have probably gotten the information he needed to make the jump, and the American government wouldn't allow him to travel because they were worried about communist influences and whatnot. So he didn't get that. Meanwhile, Watson and Crick, they were not heading in the right direction, but a co-worker of Franklin, who was a pretty shabby individual, actually, without her permission, got into her locked filing cabinet, extracted that particular photo, showed it to Crick, and he realised straight away that they were making a mistake, they were on the wrong track. And they ended up publishing in 1953 a paper in Nature. Okay, Franklin also published a paper at that time, there was another paper, a 3rd paper as well, that DNA was a double-stranded carrier of genetic information, and that the information itself was inside this helical structure, and the outside, like the outside of the staircase, was a 5-carbon sugar phosphate backbone. So the outside goes 5-carbon sugar phosphate, 5-carbon sugar phosphate, 5-carbon sugar phosphate, but the inside changes. Okay, and that was key. Watson, Crick and Wilkins won the Nobel Prize, but Franklin died of cancer at 37 due to exposure to X-rays, because as you know, the ionising radiations which damage DNA to the point that cancers can form are UV, X-ray and gamma rays. Okay, from least worst to worst, gamma being the most dangerous. So, and you don't get a Nobel Prize if you've died. You have to be alive to receive it. So she was instrumental in this discovery, but was ultimately didn't get that ultimate recognition. Yes?
Apparently, like, there were 2 or 3 Nobel Prizes awarded posthumously.
I'm not sure about that, but certainly she didn't get it. But anyway, yeah, I should look that up then. Now, here is from the lowest level of biology right up to curing cancer in remarkable ways. Here is the central dogma of biology. DNA goes to RNA, goes to protein. And the proteins have function. They have jobs, which is determined by the shape of the protein. If you change the shape of the protein, you change its ability to do that job. Now, if you change the shape, it might not, it might still do the job with the same efficiency, it might be worse at it, or it might be better at it. But that is a really key point. So here's your DNA up here. DNA does a couple of things. It makes copies of itself. Okay, that's how cells, remember, as a baby, you were only this big, very cute, and now you're considerably larger and probably not as cute. And are you the same number of cells? You are not. Okay? You started off as sperm meets egg, one cell, and when you reach adulthood, you'll be 37 trillion cells. And there's a lot of cell replication going on, but all of your cells carry DNA, except for red blood cells don't have a nucleus after a while. They just get rid of it, they don't need it. Okay, so DNA is in the nucleus. You won't be tested on this, but DNA is up here in the nucleus. Now, this job here is done by a structure called a ribosome, and this is in the cytoplasm, outside of the nucleus. DNA can't get out of the nucleus, ribosome can't get into the nucleus, so how does this information reach here? It gets copied over into RNA, and because we have no imagination, it's messenger RNA. It carries the message. So you get a copy, or actually a copy if you like, of the gene that's going to express, then the ribosome picks that up, it reads it and makes a protein. The protein forms a shape and goes off to do an important job. So we'll look at that in more detail, don't worry. But that slide 7, is fundamentally what's happening in biology at all levels. Okay, it doesn't matter if you're an amoeba or you're a redwood or anything in between. Okay, this is what's happening. You're taking this information and getting a protein out at the end. Now, very importantly, and you'll need to write this down, if I change this information, then the RNA necessarily changes, and then the protein sequence necessarily changes. And that can cause problems. And genetic disease is often along the lines, well, genetic disease is when the DNA sequence, if it's a disease, has changed for some reason, and the protein being formed is either not doing its job, and that can be problematic in its own right. You might need 2 copies of functioning genes in order to be healthy, and only 1 copy isn't enough. And so that, or the protein itself might be damaging, which is really, really bad. Okay, and there are some nasty ones where the different protein actually damages the organism that is expressing it. Now, here we go. Right, you need to be able to draw this. Okay, on a piece of paper, you always get asked this question. You need to be able to draw this. And so what does this mean? Okay, a monomer means one. The Greek part for one part, is a molecule that may bind chemically to other molecules to form a polymer, which is many parts. Now, DNA, deoxyribose acid, is a polymer. The sequences of bases on the DNA strands have this ultimately joined together to make this double-stranded genetic code for the organism. So what are the parts of the DNA strand? What are the monomers? Each monomer, 3 parts: a phosphate group, a 5-carbon sugar, and a nitrogen base. There are 4 types of nitrogen base, GATC, but the phosphate group and the 5-carbon sugar are consistent, and this is what, right down here, right spine, right there. This is a deoxyribose molecule. This is your 5-carbon sugar. This is your phosphate group. And what do you see? 5-carbon sugar, phosphate. 5-carbon sugar, phosphate. 5-carbon sugar, phosphate. 5-carbon sugar, phosphate. Repeat 3 billion times, give or take. That's the spine. But all of the information is inside. So this is like the outside of the staircase going down like that. And the inside, the steps themselves, is the information. And what do you notice about this? T always has A opposite it. C always has G opposite. And this is supposed to designate that you can't swap these in any meaningful way. These are your complementary base pairs. T always goes with A, G always goes with C. You need to be able to draw that and replicate it. Okay? And so we'll explore that a little bit. Does anyone have any questions at this stage? But that is key. This 5-carbon sugar phosphate spine, alternating repeats. Oh, another key point. The bases are always connected to the 5-carbon sugar. There are no exceptions to that rule. The bases are always connected to the 5-carbon sugar. So what you're going to have to do for about 6 or 8 marks is draw that. Okay, and label it, of course. Okay, so effectively, that's it. You need to get that right. Yes?
What does the D stand for?
Deoxyribose, which is another, that's why you get this deoxyribonucleic acid name. So the D stands for deoxyribose, or probably simpler for students, a 5-carbon sugar. And at the year 11 level, you need to show, you've got the whole class if you want. Check it one more time. Sit there, check it one more time. Okay. At the year 11 level, you need to draw the 5-carbon sugar and show where the bondings are coming off as well. Okay? So, that is key. Can I change slides?
Yeah.
Yep. Okay.
Sexy.
Okay, now, here are the actual atoms that make this up. You don't need to draw this, obviously. Okay? All 4 nucleotide monomers share a common structure, phosphate group attached to a 5-carbon sugar, which is bound to a nitrogen base. Now, these nitrogen bases, G, A, T, Z, TC, you need to remember the names. This is what adenine looks like. So this is what the monomer of DNA actually looks like. That's the phosphate group. What do you see there? P for phosphate. Okay, that's your 5-carbon sugar. Okay, this is where your connections are. And there is your adenine. There is your cytosine. There is your guanine. And there is your thymine. You'd never be expected to reproduce something like that. But that is at the level of atoms what is happening, which, if you look at it hard enough, you start to think, wow, that's kind of a bit strange. Because these little parts here in blue are what are determining functionally your eye colour, your hair colour, whether or not you grow to be 6'2" or 4'2", all of these sorts of things. How many teeth you've got in your head, whether you have 5 fingers or 11, you know, all of this is happening through those little G's, A's, T's and C's. Now, right, here we have the same information again. Now, if you really want to make a note, just on your handout, note that the base is at the first carbon position 1, and the phosphate group is at carbon position 4. If you want to take a note of that, well, that's 0, 1, 2, 3, 4. It's a 5-carbon sugar, but that's the way we're labelling it. Okay. Now, that kind of looks like a little house, if you like. But that's the 5-carbon sugar. Remember, always attached to the sugar. Okay. We call that the sugar phosphate spine, and the DNA helix consists of 2 complementary strands of DNA, each of which has a backbone and nitrogen bases tucked in the helix. Now, base pairs, complementary bases. Remember that part where I said T always goes with A, G always goes with C? Yes? Do you remember that? So, here is a strand, a little strand of DNA. Here is our T, G, C, A. Right. What is, without checking your notes, what is going to be the nitrogen base of the second strand, the complementary strand, we say? What's going to be there? A, because T always goes with A. What's going to be there? C. What's going to be there? G. What's going to be there? T. So what you are telling me, and you need to listen to this idea closely, what you're telling me is, if I know the sequence of one strand, then I know the sequence of the other strand, straight up. And that's what complementary means. They are complementary strands. They're like, not mirror images, but they are complements. If you know the sequence of one, you know the sequence of the other. G always goes with C, P always goes with A. Okay, grab one of those ones. Oh, grab one of those. You're supposed to be doing that. That's the, because you don't take it with you. No, absolutely not. That's an active assessment. Okay. Now, there you go. There is the complement, if you like. The spine is going to be the same, just inverted, and there's your T and A, G and C all the way through. And if you look really closely, there's some additional information, maybe highlight that. I did see an assessment question at one time where it asked about the bonds between the strands, and it's a hydrogen bond, which is a very weak bond. So if you heat DNA a little bit, the strands will separate into single strands, and if it cools down, they'll line themselves up and rejoin. Okay? So that's a hydrogen bond, so maybe highlight that just in case. Okay? Any questions about that? So you need to understand, when we say complementary, you need to understand that's what's going on. Well, you need, if you're given a sequence, to be able to write the complementary sequence. I think that's coming up, actually. In fact, right there. I'll give you a minute. Write that down. What is the complement of that strand? I will give you 60 seconds. It's actually a very gentle question, that one. It should take you 15 seconds, maybe. Okay, start here. What is the complement of T? A. What's the complement of A? T. What's the complement of C? G. So that's GTA, 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 GTA. Complement of T? A. Complement of A? Complement of A? T. And that's it. Okay? That should be your answer there. A nice little 1 mark multiple choice question. Okay, now. There you go. Now, if you wanted to do the complement of, by the way, watch this closely. It's going to seem like voodoo magic. Okay, want to see my one and only magic trick for the day? Yeah?
Yeah.
So, here is a gene that I want to express as a protein. I make a complement of that, which is going to be my messenger RNA, and then if I make the complement of that complement, I get my original sequence back, if you follow.
Yeah.
That's it. That's as far as the magic trick goes. Okay. You will see. By the way, the blue one you're reading the black one that way. You're reading the blue one that way, and then you read that one back that way again. So the complement of the complement is your original sequence. And the reason why that becomes important. Oh, actually, here we go. Slide 14 is definitely a cram card. I mean, almost every slide is crucial in that slideshow, but this is how cells make copies of the DNA molecules. Okay, why do cells make copies of DNA? There are only 2 reasons. When the cells are dividing, yep, and when genes are being expressed. That's it. Okay, so these are the two jobs the DNA has. Very, very important. Cell division is when you're making another cell for some reason. Maybe my skin cell's getting a bit manky and old and horrible, takes a lot of damage, so I need to replace that, and the old one falls off, dies, falls off. Okay. Cell replication. Copies both strands, making two new DNA molecules. Uses DNA polymerase. Happens when a cell splits. You don't really need to know this bit. Happens when a cell splits for some reason, as I said, when the cell is being copied. Transcription, to transcribe, is to copy over. Copies one strand of DNA, making messenger RNA. Uses RNA polymerase. Again, you won't need to know that one. Happens all the time. In your cells, your DNA in each cell is making hundreds, if not thousands, of proteins almost per second. Like, it is ridiculously fast, and it happens all the time because that's what being alive is, to express proteins. Okay? This only happens in terms of making additional cells or replacing a cell that needs to be replaced. So those are the two jobs there. Any questions? Yeah.
Why do you have to eat protein in order to be made?
Protein is broken down into base components which are then used to make you. Okay? That's what you're doing with all your foods. You've got like, okay, what is the spine of the DNA molecule made of? No. That's very specifically, we just covered it. It's a 5-carbon sugar attached to a phosphate group. So it's a sugar phosphate spine. If you're a betting man, how important do you think taking in phosphorus in your diet is? Yeah, otherwise you cannot make that sugar phosphate spine. So you better believe that phosphorus is an essential nutrient in the environment. You know, what do you think fertilizers are? What do you think they're spreading around there? Phosphorus. Yeah, get it out of rocks. You'll cover that in the rock cycle. You get it out of rocks, and as soon as it gets into living things, it gets swapped around. I eat salad, bang, 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 bang. I defecate, that gets used as fertilizer, and the phosphorus is just going through me, going back into the ground, ting, 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 like this. So it might, phosphorus as a molecule, might, as an atom, might go through 100,000 living things before it finally ends up in ocean sediment and gets buried, compressed, and becomes rock again. It might be 100 million, even 1 billion years before it gets uplifted, weathered, and back into the life cycle again. So yes, that's what's happening with digestion. Now, here is cell replication. Cell replication occurs when one strand is used as a template to make a copy of the other strand. I cannot emphasize that point enough. You need to understand this idea. The blue part is a bit of background, but you don't need to know that. Okay. Replication occurs when a DNA copy is needed, most typically during replication, and we'll look at mitosis in a minute. Now, let's have a look at this. Okay, here's DNA polymerase. DNA polymerase makes copies of DNA. Okay, helicase is just unzipping the DNA like a zipper. Okay, now watch closely. This is part of the whole magic trick thing. Here is CAGT. This is on my main strand. CAGT. Right? I'm reading this way. Now, what is the complement of T? A. So there you go. Here's the DNA helicase. What is the complement of G? C. What is the complement of A? T. What is the complement of C? G. Okay. Now, watch closely. TGAC is my original sequence. The complement of that is ACTG. Right? which actually is a copy of this strand.
Oh.
What's the sequence we were just talking about? A, C, T, G. Okay, so, well actually I've only got my, where are we? Jeez, I don't even have a lineup. Okay, but that is actually a copy of this strand down here, and this one is making a copy of that strand up there. It's using it as a template. Remember, the DNA nucleotides are complementary base pairs. If you know the sequence of one strand, you know the sequence of the other. So you use the first strand to make a copy of the other strand. And you use the other strand to make a copy of the first strand. So if you look here, if we keep going down, let's go down. What's the complement of C? G, A, T, G, C, T, A, and what have we got down here? A, C, T, G. A, C, T, G, A, C, T, G. I use this strand to make a complement of that, a copy of that strand, and that strand to make a copy of the first strand. And that, so what's happening when my skin cell or any cell makes a copy of itself, that is what goes on. Zip, and now I have this strand, which is old if you like, and this is new, and that's the old one there, and this is new, and this goes into cell 2, and this goes into cell 1, if you like, and off it goes from there, which might be the mother cell and the daughter cell. Okay? Any questions? So that is what we mean by template. I read one strand to make a copy of the other strand. That job this is not making a copy of the black one, it's making a copy of the green one. Okay, so you need to have that idea clear in your head. Now let's have a look at this. This is gene expression. How do we say genes express themselves? And what we mean by that is a protein is going to be made. Okay? And we mentioned, what was the central dogma of biology before? Central dogma of biology? DNA leads to RNA leads to protein. That's it. DNA leads to RNA leads to protein. Now, a segment of DNA is copied into messenger RNA by you don't need to know that enzyme. This occurs in the nucleus. I mentioned this before. Now, ribonucleic acid, RNA, is single-stranded, not double-stranded, and oh my glob. AGC, where's our T gone?
It's gone.
RNA does not use T, it uses U, uracil. You need to highlight that point. There are no T's in RNA. It works the same way. RNA is a very old molecule. It works the same way, but what we have here. So, what we have here, let's look at this process again. We've opened the DNA up. I want to make a protein. We've opened the DNA up. Here is ATC. What is the complement of A?
U.
U for RNA. What is the complement of T? A. What is the complement of C? G. So it's using this green one as a template to make a copy of TAG, the red one. I use the green one to make a template of the red one. Then that is used in a ribosome that goes down here. But that's your sequence of RNA. Okay? So, sorry, question or? Yeah. So how is everyone on that? I use the green one to make a copy of the red one, and I use that information to go down here and a protein gets made. Okay? Because the messenger RNA takes that information out of the nucleus to the ribosomes. Okay? Now, you won't need to know the molecule. There we go. Here's our ribosome. Now what is it doing? Look closely.
Making more.
Now, when you read DNA, it's not making DNA. This is a protein up here. So write that down. There's our protein. This is what we've wanted all along.
It's
All along.
protein in and forming it.
Yeah, well, This is our messenger RNA. Write that down too. Here's our ribosome. So there's our ribosome, there's our messenger RNA, there's our protein. Now when we read our ribosome, we don't, listen, we don't read one nucleotide base at a time. You read it in groups of 3, called a codon. Down here, 3 nucleotides. Okay, and so why is this important? What's this little doohickey doing? tRNA,
Who's just flying around?
tRNA plus amino acid. Now, watch closely. This is voodoo magic number 2. Right, now let's see. I've got to look closely. G-A-U. Okay. G-A-U, which remember is kind of like a T. What is the complement of G-A-U? C-T-A. Correct. Right. Now, this is the messenger RNA. This is the tRNA. Now, these tRNAs are being sucked in randomly. It just comes in randomly. Watch. Now, here's the simplest computer code ever. If the tRNA sequence is a complement of the messenger RNA sequence, drop your amino acid off. Otherwise, it just spits you out. So you have to have there are many kinds of tRNAs. There are 20 amino acids, and the tRNAs carry their amino acid. Now, it only drops the amino acid off if its complementary base pairing occurs here. Now, in this quite poor example, everyone that comes in finds its complement, and so it gets dropped off. But there's a lot of things that just go in, get thrown out, go in, get thrown out, fail, 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 succeed, fail, 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 succeed, 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 fail, 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 like that. That's what's happening. You have to have a match to drop your amino acid off. And then the protein comes out and off it goes from there. So you started with a sequence of G's, A's, T's and C's, and you end with a chain of amino acids which form a protein, which when the construction is finished, forms a shape, and then it's got a job, like maybe hemoglobin, which is at four proteins, 2 of one, 2 of another, that join together to form this little machine that carries your oxygen around. Yes?
What happens to the ones after they drop the amino acid?
They go and get another amino acid. And so there's this whole process going on over here, a little production line, if you like. And in answer to the question we had before, this is why a healthy diet has all 20 amino acids. Because without those amino acids, you cannot build proteins. Yeah? So the guy in Australia who is living on potatoes for a year because he wants to lose weight, that is not a healthy diet. Because he's not getting essential nutrients. Okay. Is everyone kind of with me on this? I don't expect you to immediately go, oh yeah, that makes perfect sense. But hopefully you're looking at this going, oh yeah, there has to be a matching occur. If this was random, you'd never get the exact protein sequence. It can't be random. There has to be an error checking, guys. And the error checking is at the level of codons. These groups of 3. So in last year's exam, I wrote last year's exam, I think. Or at least I wrote that question. I had a codon question, where here's a group of codons, here's a codon table. What's the protein sequence that I would get? Okay, so it was like a hands-on activity for that. But that is what is happening there. Now, here's the big question. If everything in your biology revolves around constructing proteins, and the machine that builds the proteins is a ribosome, you can't have proteins without ribosomes. What the heck are ribosomes made from? It's a chicken and an egg thing. I can't have proteins without a ribosome. But if proteins are made out of ribosomes, well, that won't work because I need ribosomes to make the proteins. Yes?
For the amino acids, how there's 20 different ones, do they each match with the sequence, or is the amino acid random to the sequence?
No, they match. The tRNA takes particular amino acids. And anyone else from my puzzler worked out what the ribosome is constructed out of? Functionally, RNA.
Oh.
So it's a machine made out of RNA and a couple other things, but functionally made out of RNA, which is just a string of, nucleotides, and then it snaps into place and you get this little machine and off it goes. Protein synthesis. Let's have a look at a video. Your poor little brains are probably a little bit tired by now. This is, okay, guys, I'll just remind you, this classroom is, the speakers, they haven't sorted them out yet, but you'll need to be quiet. Shh. You'll need to be quiet because it's actually hard to hear, but this straight away is your messenger RNA going off. I've done every volume. This is already at max.
There can be several million ribosomes in a typical eukaryotic cell.
That's a ribosome. That's your messenger RNA. Now, oh, by the way,
Machines use the mRNA copy of the genetic information.
this really critically, guys. That's the last warning you're getting. This is not a drawing or a cartoon. This is a model at the level of atoms and molecules. So this is the most accurate model we have of what's happening. So see all these differences in coloration? This is representative of individual atoms and molecules. Okay, so it's quite in depth.
To assemble amino acid building blocks.
So that's a protein coming out of the ribosome.
For life. Let's see how it works. The ribosome is composed
There's-
of one large and one small subunit that
Click.
assemble around the messenger
Ching, ching.
RNA, which then
There you go.
passes
There's
through
your
the
nucleotides.
ribosome like a computer tape.
Tink, 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 tink. Feeding in, feeding in.
The amino acid building blocks,
They're your
that's the
tRNAs.
small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA.
So when you
that
get
it
your
can be
complementary
read
match here, it drops the amino acid off there, and your protein gets longer.
On the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule.
And it's all complementary pairing.
The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site, and then ejected from the ribosome to be recycled. As synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
There you go. Now, we've got a few minutes left. Let me just check quickly. Code on. Okay. Now, okay. Just a second. I'll just hold off on questions. Now, I've got 4 minutes left and there's 2 slides I want to check, so let's just go through those and then we'll get a complete video. Right. So what we have there, here's the gene which results in the DNA sequence. There's your DNA sequence, which results in a messenger RNA sequence. Now here are your little tRNAs. There's your little tRNA, and it only has a sequence of 3, that anticodon. So I started with TAC. TAC codes for, what's the complement of T? A, complement of A. U for RNA. What's the complement of C? G. So that's my first codon. Now if I get a match, a complementary match, which is UAC, that tRNA will drop that into a protein. And so that's what's happening. You get your DNA, goes to RNA, your RNA is tested against tRNAs carrying amino acids, and if you do get that match, that complementary base pair match, it gets added to your protein sequence. And that's how your DNA gets a very precise protein sequence at the end, and that protein will have an important job to do. Now, importantly, we'll look at the videos later, but this is the last key slide. Mutations. Change that occurs at the level of DNA that may add, insert, delete, or rearrange genetic material. That sequence, the gene sequence, is very, very important. If you change the gene sequence by even a little bit, you'll change the protein. And some of those changes are fatal, invariably fatal. Some of them, it doesn't really affect the organism. Remember, you have 2 copies of each gene, and some of them, but that change may affect offspring, and some of them might improve the organism a little bit. So the mutations can be important. It happens at all genetic levels, just at nucleotide bases, sometimes whole chromosomes. You get extra copies of them. Trisomy 21, where you get an additional chromosome 21, causes some problems. You've got to have an extra piece of genetic information there, okay? Or sometimes you can lose chromosomes, okay? Chromosome fusing is very interesting. 2A and 2B fused in humans. So if you understand genetics a little bit, all apes have 48 chromosomes, except for humans with 46. And wait. And so people used to look at that and go, that's kind of interesting. So they went looking, and sure enough, they found a joining site in humans in chromosome 2, which is quite large. No, don't pack up. Chromosome 2 is quite large, and it consists of 2 chromosomes that are fused together, 2A and 2B. Otherwise, humans would have 48 chromosomes, like every other ape. Now, stop. The causes of these errors are DNA replication, when cells are being multiplied, radiation from the sun or X-rays, or possibly chemicals. And if that damage isn't repaired, then you have a mutation, which may cause problems in its own right. Well done, everyone.